అలా అక్కడికెళ్తావు కదా నువ్వు స్నానం చేసి అక్కడ తిని పెడతారు. తెస్తారు తిను.
Hello everyone. We'll wait for some 5 minutes for everyone to join and then we'll start the session. Okay, so we'll start the session now. Can someone confirm that I'm audible?
Yes, ma'am. Able to hear your voice.
Okay, so welcome to this extra TA session for drug delivery principles and engineering. We'll just go through all the topics once again and we'll discuss few extra questions that I had. And yeah, that's all we'll do today. I hope your problem was solved last time, the progress thing that you were mentioning.
Ma'am, no, ma'am. Now the course progress same 67.32% is there only.
I
But
see.
I have in the unit wise progress also only 2 weeks I got the 70%. The remaining all the weeks I got 100%. And the next assignment score is also only 2 weeks I got 70%. The remaining all weeks I got 100%. I listened all the recorded classes.
Right, right.
But same, ma'am,
Uh,
last 4 weeks onwards, the course progress is not changing, ma'am.
okay.
4 weeks onwards, I think so, 4 to 5 weeks.
Yeah. So I think this is not only your problem. Few other people are also there who are facing the similar kind of problem, that they have completed all the sessions but still the progress is not increasing. So you had mailed to Kavita, right? I think she is
Yes,
the
ma'am. Yes, ma'am.
yes, she is the technical in charge and she is handling it, I guess. But whoever is facing this
Yeah,
problem, just notify them that you are facing this problem and they are working on it. They'll uh, clear this out. So don't worry about it.
ma'am. Okay, ma'am. Thank you, ma'am. Ma'am, it will shows any impact on our certificate.
No, 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 no. So this is the technical issue, right? This is not from your end. This is something that was
Yeah.
from their end. So yeah, this won't affect your certificate. Just notify them whatever problem you are facing, and this technical issue will be resolved quickly.
I have complained to the Rajesh Yadav sir
Yes, yes,
also,
yes.
ma'am. I have made.
Yes, I know. I talked to him, and he only told me that Kavita is the in charge. They have also notified Kavita, and she only does this technical changes, so she'll be doing this. So
Okay, ma'am.
this interface, she's
Thank you, ma'am.
handling this interface. Yeah, but
Okay,
you
thank you so
do need
much,
to
ma'am.
notify. I think you have notified, but others also, whoever is facing problem, just mail them so that they know that you are also facing this problem, and it will get resolved. Yeah.
Good evening, madam.
Yes.
Okay,
Good
ma'am.
evening.
Okay, thank you, ma'am.
Yeah.
Madam, actually, I was missed one of the assignment, week 3 or week 4.
Okay.
So, is there any further chance to write that assignment exam?
Oh, I don't know. I'm not sure that's possible, but you can try mailing them that you missed an assignment. Is it possible? Yeah.
Okay, okay, ma'am. Then, what about that October 29th? We are having the exam like that previously. I know that one, but till now we didn't get any information regarding that hall ticket, whatever it may be.
Yes, yes, that also I talked to them, Rajesh, and they also don't know when the hall tickets will be released. So it's kind
Okay.
of NPTEL's thing. They haven't released the hall ticket. So yeah,
Why
you guys
why
have to
we are
wait.
asking means now holidays and we are busy in college works.
Yeah, yeah, I
Not
understand.
where if I missed that one, there is no chance again to write that exam.
Sorry.
If I miss it to see that mail or hall ticket, whatever it may be, if
Hmm.
once the exam date was finished, it means it is difficult to write the exam.
Right, right.
So
Uh,
for that I'm
I understand, but it's not in our hands. So the NPTEL people will send the hall tickets. It's all in their hands.
okay, madam. Thank you.
Yeah. And also, you guys were all asking what kind of questions will come in the final exam. So I think multiple choice questions will only come. So you just need to go through all the lectures in details and like assignments. Not it might not be similar question to that we discussed in the assignment. The same question, but it will be pretty similar to what we all did in the assignment. So just go through all the numericals and all the questions, sorry, all the lectures, and just pay attention to the details and you'll be fine. Okay, so I'll be now sharing my screen. Let me know if you guys can see. Okay, I hope everybody can see my screen.
Yes, ma'am. Yes, ma'am.
Okay, so as I said, it will be multiple choice questions, and so these are all the topics that that were covered so far. In week one, pharmacokinetics, prodrugs, and polymer synthesis and properties were discussed. In week two, biomedical polymers, polymer drug conjugates, diffusion control system, controlled release reservoir systems were discussed. So in week one, some of the there were some equations for first order, second order, and elimination equations. And in week two also the controlled reservoir systems also had some equations. So please try to remember these equations. This might help you in the exam. Week three, controlled release reservoir systems, non-erodible systems, and non-erodible and erodible systems, and hydrogels. And in week four, nanoparticles, nano and microparticles, liposomes. So here also the different kinds of methods of preparation, such as single emulsion, double emulsion, how they are actually being prepared, and if they are being prepared, where exactly the drug is getting encapsulated in case of liposomes, or in single emulsion or double emulsion particles. These things you need to pay attention. Then in week five, protein adsorption, tissue engineering. In week six, tissue engineering again, and drug delivery in tissue engineering and implant associated infections were discussed. Further, in week 7 and week 8, most of the route specific delivery, such as oral, subcutaneous, intramuscular, transdermal, inhalation, buccal, rectal, intraarticular, intravenous administrations were discussed. So, go through all the advantages and disadvantages of these route specific deliveries as well. Then week 9, immune and blood response to materials, immune system, complement system and blood clotting and hematocompatibility of materials, adaptive immune response and vaccine. And in week 10, vaccines, immune isolated cell therapies, isolated cell and gene therapy. And in week 11, all gene delivery with vectors, polymers, gene as vaccines, cancer vaccines, immunotherapy was discussed. And in the last week, responsive drug delivery systems, targeted drug delivery systems, and nanotoxicology and translation pathways were discussed. So you just need to go through all the lectures and pay attention to details. So from the lectures, I just took a few portions just to highlight how to pay attention. So here in the beginning of week 1 or week 2, lipid water partition coefficient was discussed, right? The higher the lipid water partition coefficient, the greater rate of transfer across the membrane. So polarity of a drug, by increasing the ionization, there will be increased polarity of the drug. By increasing the ionization will decrease the lipid water partition coefficient. Okay, so lower so this will lower the transfer across the membrane. And if there is decrease in polarity of a drug, suppression of ionization will be happening, and there will be increased lipid water partition coefficient. So sometimes so they might give a hypothetical question that you have a polar drug, what will be the rate of transfer, or they might give two three drugs based on their polarity. How will they what will be their rate of transfer across the membrane? So you can expect such kind of questions, although it will be multiple choice questions, but it might be tricky. Then here is another thing, step reactions and chain reaction was discussed. in the lecture. So here this is what they mentioned, that monomer consumed rapidly in case of step reaction, while molecular weight increases very slowly. Whereas in chain reaction, molecular weight increases fast while monomer consumption is low. So what is basically happening is, in case of step reaction, if there is one, then it can immediately react with another to form a dimer. And if there are two, then they can react with another to form a trimer. But in the chain reaction, it can only happen if there is one, and it can only react with another one that will grow to form a dimer. And this dimer can only react with another monomer to form a trimer. But here, this dimers can react with monomer, or the single dimer can react with another dimer to form a tetramer. So there is a lot of combination can happen here in case of step reaction. At a point, they will have different combinations, dimer, trimer, tetramer, but here only a single combination will be there. So if they ask that what will be the which of these will have a molecular rate at a particular time, if they are using a similar polymer and they are using one by step reaction, another by chain reaction. So here the correct answer would be the molecular weight will increase only in case of chain reaction. Okay. I hope I'm still audible and everybody is able to see my screen.
Yeah, ma'am.
Yes, ma'am.
Okay. So these kind of details you need to uh, pay attention to. Then polymers, about different polymers was discussed. So you need to go through all the advantages, disadvantages of these polymers and the structures as well, rate of degradation of different polymers. There was also I think bonds, what kind of bonds degrade faster than others. These kind of details you need to pay attention to. So here PLJ was discussed, which is the mixture of PLA, polylactic acid, and polyglycolic acid. So here it is mentioned that PLA hydrolysis is slower than PGA because of bulky methyl groups, and PLGA hydrolysis depends upon composition. So so this is PLA. As you can see, here is a bulky methyl group, and this is PGA without any methyl group. So this PLA is more hydrophobic compared to PGA. So this will prevent degradation, uh, faster degradation, whereas PGA will uh, lead to more higher rate of degradation. So if suppose they say, so PLJ is composed of both PLA and PGA, right? Can you, in the middle, can you at least say yes or no? So I'll know you guys are still online. Hello?
Yes, ma'am.
Yes. So so PLA and PGA is PLG is a combination of both PLA and PGA, right? So if there is the 50% of PLA and 50% of PGA, the degradation will be different than when there is 90% PLA and 10% PGA. And when there is 90% PGA and 10% PLA, right? So higher the percentage of PGA, more will be the rate of degradation, and higher the percentage of PLA, lesser will be the rate of degradation. So sometimes the question might come like this as well, that these are the different compositions of PLGA with different polymers of PLGA with different compositions of PLA and PGA, and which one will have a higher rate of degradation to so this is how, if you pay attention to details, you'll be able to solve these kind of questions. Okay, so here is a question. Let's solve this. Which of the following is not a polymer? Antibody, albumin, cholesterol, dextran, collagen, or pullulan? Would someone like to volunteer?
Ma'am, antibody.
Okay.
Antibody.
What about others?
Maybe antibody, ma'am.
Okay. Okay. Yeah, I think all of you want to select antibody, but okay, here it is asking which of is not a polymer. But you all know that antibody is a protein, right?
Yes, ma'am.
Yes. And protein is a
Polymer.
right.
Polymer.
Right. So will antibody will be the correct answer?
No, ma'am. Sorry.
Next, albumin. You can say it is another protein. So this is also going to be in the category of polymer. Dextrin, collagen, and pullulan, these are also some kind of polymers. But if you see cholesterol here, it is a single molecule. So cholesterol will be the correct option here. So yeah, so here I wanted to
Yes.
say, sorry, yes. Was someone trying to say something? Hello?
No,
No,
ma'am. No,
ma'am.
ma'am. Continue.
Okay. So yeah, so you guys selected antibody because we all know the Y shape of the antibody and it feels like it is single molecule, right? But actually if you look inside, it's a polymer, it's a protein. So these kind of details you need to uh, follow when you are answering these. The questions might come tricky like this. Okay, so
Yes,
here
ma'am.
cholesterol will be the correct answer. Next, I think this is a very easy question. The study which deals with how the drug interacts with body via absorption, metabolism, elimination, etc. is called.
A.
Pharmacokinetic.
Pharmacokinetics.
What about others?
It's pharmacokinetics.
Okay. Yes, you guys are all correct. It's pharmacokinetics. So here the question was straightforward. So sometimes these kind of straightforward questions might also come. So what I'm trying to say is you have to go through the entire lecture properly. And just because I'm saying pay attention to details doesn't mean that you forget about the simple things that are also there. Now, this was another detail based question. Which of the following bonds have the slowest erosion rate? I think this question was also there in one of the assignments. I don't remember, but yeah. Anybody would like to answer?
Anhydrates.
Okay. What about others? Anybody else? Yes, anybody? Okay, so here the correct answer will be amides. So the rate of degradation is anhydrides degrade faster compared to esters, and then comes amides. Amides have the slowest erosion rate. Okay. Okay. So if you go through the lectures at one point of time when they're discussing all the polymers, different kind of polymers, there it is mentioned that anhydrides has a higher rate of degradation compared to esters and then amides. Okay, let's see the next question. So you have attached PEG to a polymeric nanoparticle. What is true for the resulting conjugate? I'll give you some time to go through all the options. Let me know what you think the answer is.
First option.
Okay.
Fifth option, ma'am. Long.
Okay. What about others? Anyone else would like to answer?
Ma'am, third one, pegylation of nanoparticles
Okay.
conforms to the new identity requiring FDA approval.
Okay. So here there will be two options that will be correct. First is pegylation of the nanoparticle confers a new identity requiring FDA approval, and the entity will also most likely have a longer residence time in blood. So I'm not sure whether they'll deliberately mention there that you have to select multiple correct answers or not, but sometimes the questions might also come like this, that uh, select which of these are true, or what is true for these such and such polymer, or whatever question is there. So you have to think what all options can be correct, and if there is more than one option you feel that is correct. If you feel that more than one option are correct, then you should definitely go for it. Okay, so here both pegylation of nanoparticle confers a new identity requiring FDA approval, and the entity will most likely will have a longer residence time in blood. These two options will be correct for this peg-attached polymeric nanoparticle. Okay. Here is the next question. Liposomes are generally formulated via which of these methods?
Ma'am, first option, solvent evaporation and hydration.
Okay.
First option.
Okay. Anybody else would like to answer?
Yeah, ma'am. First one, solvent evaporation hydration.
Okay. Yes, for this you guys are all correct. It is solvent evaporation and hydration. Okay, these were all the questions, few questions I had today. And yeah, let me know if you guys have any other doubt or want me to repeat what I said again. But you need to go through all the lectures very carefully. Um, anything, they can ask anything. Anybody has any doubts?
No, ma'am.
Okay. What about others?
Ma'am, what about hall tickets?
Yeah, I already mentioned it's in the hands of the NPTEL people. They'll give you the hall tickets. We are not sure when they are going to give it to you. Passing criteria. I'm not really sure. I think you can go in the main website. You can see it there. For the drug delivery course, it might be there. What is the passing criteria? I'm not sure how much percentage you need to pass the exam. Okay. Okay. Do you have any other doubts from the course itself or from any of the previous assignments? Or do you want me to go through these questions again?
No, ma'am.
Okay. What about others?
Ma'am, roughly how many questions do we expect from the previous assignments?
How many questions? So
Yeah.
the questions might not be the same as the assignments. Assignments was just to give you a glimpse of what kind of questions will come in the exam. Uh, in the final exam, the questions might be different. Yeah.
Okay.
But you can see for a particular topic how they are forming the questions. From there, you can gather that what kind of questions you can expect in the final exam.
Ma'am, total how many questions will be there?
I am not sure. I don't know.
Ma'am, can we search in the Google the last time how the question paper will be there?
You can try. I don't know if it will be accessible
Yeah.
because these are offline exams, no? Like offline, I mean, you have to go to the center and give it. So
Yeah.
I'm not sure if it will be available. But you can
Okay.
try searching for previous assignments and more. If it is available, then you can try solving those as well.
Yeah, ma'am. Thank you.
Any other doubt? I think they won't allow your phone to give the exams. So yeah. Um, I don't think negative markings will be there, but for some questions, if it will be, then it will be mentioned in the question itself. But as far as I know, there is no negative marking. Okay, any other question? Or any doubt? From which week? I can't specify. There is a lot of information uh, out there, so I can't really say from which week you can come. Questions can come. But as I showed you in the presentation, like from every part, there are some sections where more information is given. You can focus on those if you want to solve maximum questions. But if you ask me, I would say uh, the MWMN, the number weight, molecular weight, and weight average molecular weight, these kind of numericals will come, might come, and uh, the polymers, different kind of polymers related to those. And I'm yeah, but for other weeks, you have to go through all the lectures. The questions can come from anything and anywhere. But the numerical parts, you can whatever sample problems was discussed in the assignment as well as in the lectures, throughout the lectures, you can try going through those numericals and equations again and again. Similar kind of numericals you can expect in the exam. You can score in the numerical part if you know about what the equation is asking, what kind of equation it is demanding.
Ma'am, from the numerical side, they will ask the same question, same the numericals, or otherwise they're having the same model and they will change the numericals.
Yes. They might not ask the same question, or might ask that, I don't know, but similar kind of model you can say. For example, the erodible or non-erodible systems were there, the time of what will be the release time or thickness of the system, those things, those questions we solved, right? There was a particular equation for that. So that you can,
Yeah.
that is how you can solve those equations.
Thank you, ma'am.
Yeah. Any other question? Okay, anyone has any other doubt?
No, ma'am.
Okay. What about others?
No, ma'am. No in doubt.
Okay. If you don't have any questions or any doubts, you can leave the session. And those who are joining now, I'll repeat again what we discussed till now.
Thank you.
Welcome. All the best for your exams.
Thank you, ma'am.
Thank you, ma'am.
Welcome.
Thank you, ma'am.
So whoever is present now joined late, is it? Shall I repeat what we discussed till now?
Yes, ma'am.
Okay. We'll wait for a minute or two because some people are still joining. Then we'll start again. Is that
Sure, ma'am.
fine?
Yes, ma'am.
Okay. Okay. So when we started, I think in the previous sessions, people were asking what kind of questions will come. So MCQ type questions will only come in the exam, and it might be one option or multiple options correct type questions. It will be mentioned there in the question, or sometimes it might not be mentioned. So you have to look for all the correct options. And yeah, and if any of you are facing problem for the progress thing in your portal, uh, NPTEL is handling that. Kavita is the technical in charge, I think. You can mail if you are facing any such problem and it will be resolved. Also, about the hall tickets, uh, that is also handled by NPTEL. We are not sure how when they are going to send you these tickets. So you just have to wait or you can write to them. Okay, I'll now share my screen. Let me know if you guys can see my screen. I hope it is visible to everyone.
Yes, ma'am.
Okay. So first, I just went through uh, all the topics that were discussed so far. So in week one, pharmacokinetics, prodrugs, and polymer synthesis and properties were discussed. So if you go through this first week, I think there were some graphs or plots for these pharmacokinetics as well as the drug efficacy and those things. So pay attention to those kind of information when you are going through these lectures. Uh, Also polymer synthesis and properties, the step reaction, chain reaction, as well as the number average molecular weight and weight average molecular weight. Those equations, keep in mind that such kind of questions might come. And in week 2, biomedical polymers, polymer drug conjugates, diffusion control system, control release reservoir systems were discussed. So here also different kinds of polymers and their properties was discussed. Uh, so a lot of information based questions, and in case of control release system, reservoir systems, some equations were given. So some numerical based questions might come from these topics. Then in week 3, control release reservoir systems and non-erodible systems, control release non-erodible and erodible systems, and hydrogels are there. Again, some information from hydrogels and some numerical kind of questions from these control release might come. And in week 4, nano and microparticles and liposomes, their synthesis method of different kind of synthesis procedure and procedures were discussed. So you need to go through those procedures, uh, the methods, and how exactly the drug is getting accumulated and where exactly the drug is getting encapsulated. Pay attention to those things. For week 5, protein absorption and tissue engineering, and week 6, drug delivery and tissue engineering and implant associated infections. You can simply go through the lectures, and some information based questions might come from these week 5 and week 6 topics. Then in week 7, route specific delivery such as oral, subcutaneous, intramuscular, transdermal. Also in week 8, again inhalation, buccal, rectal, intraarticular, intravenous administration, approved nanocarriers and immune system. These all route specific deliveries and their advantages and disadvantages were discussed. So pay attention to those advantages and disadvantages, and yeah, questions might come related to that. I hope I'm still audible.
Yes, ma'am.
Okay. In week 9, immune and blood response to materials, immune system, complement system, and blood clotting, hematocompatibility of materials, adaptive immune response, and vaccines were discussed. These are all information based. And then week 10, vaccines, immune isolated cell therapy, immune isolated cell and gene therapy. And in week 11, gene delivery vectors, gene delivery polymers, genes as vaccines and cancer vaccines, immunotherapy was discussed. And in week 12, responsive delivery systems, targeted drug delivery systems, and nanotoxicology and translation pathways were discussed. So in week 10, 11, and 12 also, it was all information based. You can go through these lectures and pay attention to details, and multiple choice questions might come from these as well. So just to give you a glimpse what kind of question might come and how to pay attention to details, I'll go through some parts from the lectures and then we'll discuss some questions. So here the lipid water partition coefficient was discussed in the initial lectures, where it says the higher the lipid water partition coefficient, the greater the rate of transfer across the membrane. So, and how this lipid water partition coefficient is affected. So, greater the polarity of a drug, by increasing ionization will decrease. Uh, by increasing ionization will decrease the lipid water partition coefficient. So basically the increased polarity will decrease the lipid water partition coefficient, and by decreasing the polarity there will be suppression of ionization that will increase the lipid water partition coefficient. So basically more transfer will occur if the drug is more if the drug is less polar, and if the drug is more known if the drug is non-polar, it will have a higher rate of transfer, versus if the drug is polar, it will have a lower rate of transfer. So questions might come like different drugs with based on their polarity might be arranged, and you might get asked that what will be the rate of transfer of these different kinds of drugs. So based on these fact that an increase in polarity leads to decrease in lipid water coefficient, and decreasing polarity leads to increasing lipid water coefficient, you can rate the rate of transfer of different kinds of drugs. So paying attention to these kind of details when going through the lectures can help. Further, this was again we were discussing about step reaction, chain reaction. So what I'm trying to say is that questions will not come from these parts, but when you are going through the lectures, you can focus on these parts like this, like how I'm focusing right now while discussing, so that you can answer the questions more easily. So in step reaction and chain reaction, it was mentioned here that monomers consumed rapidly while molecular weight increases slowly in step reaction, whereas in chain reaction, molecular weight increases fast while monomer consumption is low. So what they mean at this point is, so monomer is consumed rapidly. That is, if initially if there are single monomers, more and more monomers will interact with each other to form dimers, and then there will be monomers and dimers, so more dimers can interact with each other dimers, or they can interact with single monomers to form trimers as well as tetramers. So these single molecules or dimers or trimers get consumed very rapidly. But if you see the overall molecular weight, it is again just a dimer or monomer, and the overall chain length of the polymer is not increasing, so the molecular weight will increase very slowly. Whereas in case of chain reaction, if you see, only it will form step by step. If there is monomer, it can only interact with another monomer to form a dimer, and if there is the dimer, it can interact with another monomer to form a trimer. So it will go stepwise, that first monomer will come, then dimer, and then trimer. So it will slowly increase the polymer chain length, and then slowly the molecular weight increase really fast as compared to the step reaction. So if suppose there is a question that at some point of a similar kind of polymer was formed by one by using step reaction and one by using chain reaction, but at any particular time, which of these will have a higher molecular weight? So you can say the chain reaction will have a higher molecular weight, because this might be still in the initial points of trimers or monomers, but the chain if the chain length has increased, the chain this will have a chain reaction will have a higher molecular weight. Okay, I hope I'm still audible. Am I still audible and you guys can still see my screen?
Yes, ma'am, you are audible.
Okay. Okay, so next we are discussing about polymers. So different kind of polymers was discussed, and their properties and how they are used, and their rate of degradation, how it is based, all those things, and different kinds of bonds and how they uh, affect the rate of degradation was discussed. So here is an example. So PLGA is a polymer. So PLGA, polylactic glycolic acid, is a polymer that is formed by both PLA and PGA, that is polylactic acid and polyglycolic acid. So if you go through the lectures, you'll come across this small sentence, the PLA hydrolysis is slower than PGA. So PGA degrades faster as compared to PLA. So why that is happening is because bulky methyl group. So if you look at the structures of PLA and PGA, this is PLA. PLA has a methyl group. So it is kind of more hydrophobic, and it will interact less with the water, whereas PGA, it will interact more with the water and leads to more degradation. So you can say when PLGA, which is composed of both PLA and PGA. So for example, question might come that there is 50% PLA and PGA and 90% PLA and 10% PGA, or 10% PLA and 90% PGA. These kind of different polymers are there. How will you uh, rate their how will you rate their rate of degradation, which will degrade faster and which will degrade slower? So you can use this fact that PLA hydrolysis is slower than PGA to rate this. So in case of polymer where PLA has a higher percentage, as I said, 90% or 80%, whatever. If PLA has a higher percentage in the PLGA, then it will degrade slower. But if it is PGA, then it will degrade faster. So you need to pay attention to these kind of details when going through the lectures. So here comes the questions. We discussed some 5 questions. I'll go through them one by one. So this was the first question. Which of the following is not a polymer? What do you think the answer is? I'll give you some time to go through all the options.
Cholesterol.
Okay. What about others? Anyone else?
Cholesterol, ma'am.
Okay. Anybody else would like to answer? Okay. So cholesterol is the correct answer. Uh, then I don't think I need to discuss much. Antibody, albumin, these are all proteins. Proteins are polymers. And dextrin, collagen, pullulan, these are all polysaccharides, polymer. These are also polymers. So cholesterol will be the correct answer here. This was the second question. The study which deals with how the drug interacts with body via absorption, metabolism, elimination, etc. is called as?
Pharmacokinetics.
Okay. What about others?
Yes, ma'am. Pharmacokinetics.
Okay. Anyone else? Okay. Yes, you guys are correct. It is pharmacokinetics. So if you go through the lectures, you'll know that in the beginning of the lectures itself, this sentence was mentioned that the study which deals with how the drug interacts with body via absorption, metabolism, elimination, etc. is called pharmacokinetics. But you won't find question like this, which of these is not a polymer. So you have to think more in uh, depth when you are going through the lectures. It might not be straight from the lectures, but you have to use whatever information you got from the lectures and use it to solve these kind of questions. Okay. This is another question. Which of the following bonds have the slowest erosion rate? Anyone?
Option C is amides.
Amines. Okay. What about others? Anyone else? Yes, you are correct. It is an amines. If you go through the lectures, somewhere it is mentioned that anhydrides degrade faster compared to esters and then amines. So the slowest erosion rate will be of amines. Here's the next question. So you have attached PEG to a polymeric nanoparticle. What is true for the resulting conjugate? I'll give you some time to go through the options. Let me know what you think the answer is. Anyone ready to answer?
I think answer C.
C. Okay. What about others? Anybody else?
C, ma'am.
Okay. Okay. Uh, you guys are correct. C is one of the
It
options
might
that
be
is
fifth
correct.
also, ma'am.
Sorry,
It might be fifth also.
fifth. Yes. Uh, you're right. I was about to say that. So here you can expect. Uh, so in these kind of questions, you can expect multiple options to be correct. One, that is pegylation of the nanoparticle confers a new identity requiring FDA approval, as well as the entity will most likely have a longer residence time in blood. Both of these options are correct related to PEG conjugation to polymeric nanoparticles. So here both of these options will be, you have to select both the options to get the correct answer. Okay, this is the last question. Liposomes are generally formulated via which of these methods? Okay. Yes, option A is correct, solvent evaporation and hydration. So yeah, these were few questions. That these are all the questions that we are going to discuss today, and also so in the assignment and in these kind of in the questions that I discussed today, these kind of questions, not the same questions, but these kind of format and questions you can expect in the final answer in the final exam. And yeah, go through all the lectures in details and also the numericals. The format might be same for the numericals as well. Whatever was discussed throughout the lectures, similar kind of questions, and also in the assignments, those kind of similar kind of questions. Uh, yeah, go through all these things carefully, and you'll be fine. Okay, any doubts or any other questions you have? Yes, anyone? Or do you want me to discuss any question or any topic again? I'm still audible, right? Hello? Am I audible?
Yes, ma'am.
Yes, ma'am.
Okay. So do you guys have any doubts?
Ma'am, is there any major impact on exam related to course in progress?
No, no, no. So you are saying you are facing problem that you have submitted everything, but your course progress is not showing higher, is it?
Exactly, ma'am.
Yes. So no, this is not only your problem. Many people are facing similar kind of problem, that they have submitted all the assignments and everything, but course progress is very low. So this is the technical issue, and you can mail it to Kavita, I think. She is handling this, and this is not yet resolved, but it will be resolved soon. But do let them know that you are facing this problem.
Ma'am, how to mail them? He should mail on the website.
It will be, I think, in the NPTEL portal or somewhere. If not Kavita, you can mail one of the TAs. They'll forward it to her as well. So one of
We can mail to an MPTL.
the course you can mail to the TAs related to the course. They can forward it to them. So in this session only, previously someone was there. So they were saying that they had got the mail ID for Kavita, and Kavita is actually the technical head, you can say, uh, for these portal interface that you are having. So she handles this technical issues. She's from the NPTEL team, so you can directly mail her or one of the TAs. But do let them know that you are facing such problem.
Ma'am, how to approach their mail ID?
I think it will be there when you open your NPTEL course.
Ma'am, directly to Kavita, ma'am.
Uh, that I'm not sure. I think she got the email from one of the TAs. But you can try looking for the NPTEL technical head team online. I'm not sure how she got it. But if you're not getting Kavita's mail ID, then you can mail to one of the TAs.
Okay, ma'am.
Can you ask to ma'am uh, to Kavita ma'am from that regarding the issue course progress?
I am not in contact with them. They are from the NPTEL's team. So I have talked to other TAs, as I said. They have forwarded their mail to Kavita. So you can talk to
Okay ma'am.
Kavita or mail to the TAs, and they'll forward it to Kavita, and she'll handle it.
Okay ma'am. Thank you.
Yeah. Anybody has any other question, any doubts, or wants me to discuss any part again? If not, all the very best for your exam. Also, the hall tickets, that also we don't know when you are going to get it. The NPTEL team is handling that. So, yeah, all the best for your exams. If you don't have any doubts or any other questions, we can close this session here itself. Or if you want me to repeat any question or have any doubt or want me to discuss any topic, you can let me know.
Ma'am, are there major chances of assignment questions to come in exams
Uh, I
or related to assignment questions? Similar.
related to, I would say it will be pretty similar to assignment questions. The kind of format was there in the assignment, similar kind of numericals, or from a particular topics, the kind of questions you got in the assignment, similar kind of questions you might get. But same question, I don't think so. But you might, if you're lucky, you might get same question as well from the assignment.
Ma'am, the pattern of questions.
Yeah, pattern will be similar to the assignment, same kind of questions, similar kind of questions.
Then we hope to be lucky, ma'am.
Okay, anyone else?
No ma'am.
Okay. Shall we close this session then?
Yeah ma'am. Thank you so much ma'am.
Thank
You're
you so much,
welcome.
ma'am.
All the best for your exams.
Thank
Thank
you,
you,
ma'am.
ma'am.